Chapter 14, Section 10, Anaerobic ammonia oxidation, also known as the Animox reactions. Animox, acronym anaerobic ammonia oxidation. What we're talking about here is a set of obligate anaerobic bacteria are going to take ammonia and nitrite and are going to be releasing nitrogen gas and in the process also producing some metabolic water. Brocardia is an example of genus found in the group Plantomycetes. Plantomycetes are a special type of prokaryote in that they do have some membrane-bound compartments within them. These are not vacuoles, these are not, organelles. This is a special type of membrane that encloses a region. Within that region, those regions, you're going to find the enzymes for Animox activity. These are referred to as Animoxosomes. Okay. The energy produced by doing this is what's going to power the CO2 fixation, but instead of using a Calvin cycle, what they're going to use is a reductive acetyl-CoA pathway. Basically going to fix two carbons together, attach it to coenzyme A, and remember that can either be reverse engineered into pyruvate, gluconeogenesis, or then fed into the citric acid cycle, producing any of the amino acid derivatives, which are the intermediates of the citric acid cycle. So, these Animoxosomes you see here are not true organelles. They house and are there for one and only one thing, these Animox reactions. Their membranes are different. I know in the last section at the end there I said that, oh my gosh, the organelles, blah, blah, blah. What I want to point out is the special lipids here, laterine lipids. Okay, laterine lipids, because of their formation, the way they're structured, remember structure function, they limit diffusion across. So whatever's inside the Animoxosome is stuck inside the Animoxosome unless it's transported out. Speaking of transport, notice here we have an electron transport chain. It's not shown. Again, it's going to be a bridged version of, and what it's doing is it's taking hydrogens from the cytoplasm and pumping them into the Animoxosome, which, contrary to the previous, I don't know, however many sections and however many chapters, we've been looking at things that have pumped the hydrogens across the cytoplasmic membrane outside the cell when we've been talking, prokaryotes. So here, cytoplasm inside this membrane-bound region known as an Animoxosome. Now, the electron transport chain, notice it's also not utilizing anything in the cytoplasm to power it. No, it is being powered by the, hydrazine, N2H4, being broken down to nitrogen gas. The hydrazine dehydrogenase, which is removing the 4 hydrogens. Here they are released, so here's 4 hydrogens inside the Animoxosome. 4 electrons are being stripped off. Those 4 electrons are going to be bled of their energy to pump hydrogens in, which is good, because some of those hydrogens are going to flow out through ATP synthase, ATP to be utilized in the cytoplasm. But notice there's no mention of NADHs here, NADHs. That's occurring out here in some other metabolic functions. Here, these 4 electrons that are being bled off, they're not going to oxygen, because remember this is an anaerobic cell. What they're being used is they're being fed back into this Animox process, because we're starting with nitrite to what is this? Nitric oxide, to hydrazine to nitrogen gas. Well, look, here I just talked about the hydrazine to nitrogen gas. 4 hydrogens pulled off, 4 electrons pulled off. Well, here, nitrite to nitric oxide. 1 electron, 2 hydrogens are being fed in. Nitric oxide to the hydrazine. Well, 3 electrons, 2 hydrogens being fed in. 1 plus 3 is 4. 4 hydrogens. 2, 4. So the hydrogens that are being pumped in basically are the ones that are flowing back out. Because the 4 hydrogens that are pulled off here and the 4 electrons are fed into the next round of this animoxic pathway. Now, technically, yes, if you look at the definition, these Animoxosomes are technically an organelle. It's a membrane-bound region that's housing certain things, blah, blah, blah. Special lipids called laddering lipids, because of their structure, like I said, they prevent diffusion. And here's those reactions I just went over. Why do we want a membrane that prevents diffusion? Well, because certain of those intermediates are toxic. Hydrazine, toxic. You want to keep that in the animoxosome so it doesn't leak out into the cytoplasm, interfering with other metabolic activities. And then here's those reactions that I just discussed on the previous slide. The nitrate that Riccardi is going to be using, this is actually provided by other microbes, aerobic ammonia oxidizing prokaryotes. Okay, other bacteria in that same environment are taking ammonia and ultimately making nitrite, as we talked about the last section. And that nitrite then can't further metabolic, reduce it or oxidize it, so they just excrete it out. Excrete it out where these Animox bacteria can then utilize it. What's interesting is even though they're, anaerobic, they can grow in the presence of some oxygen. High levels of oxygen inhibit Animox metabolism, and they favor the nitrification. Other microbes that are going to be using, the nitrite to form nitrate and certain things like that. Low levels of oxygen instead of nitrate to nitrite, nitrite to nitrous gas via Animox bacteria. Again, we're looking at these nowadays, in the past decade or so, as a means to aid in wastewater treatment. I got to clean up all the wastes coming out of the big cities. These mega cities we now have and are developing produce a lot of waste, and we need to get rid of it. We can get rid of solids, but it's the individual molecules of ammonia, nitrate that we can't just filter out. And yes, we could chemically treat them, but then the chemicals we treat with also have their side effects. So if we can use bacteria, prokaryotes, that we seed into a pond, a wastewater treatment pond that will do these conversions, and then ultimately all you get is a bunch of bacteria settling out because the cells died, and then a bunch of nitrogen gas being released into the environment. Happy, happy.